Well, the limitless potential of molecular scissors, as the name itself implies, they're used to cut DNA for intended gene targets. Well, their potential in the realm of medicine is enormous, opening doors to new treatment methods for incurable chronic and degenerative diseases. For this week's IT and Science Front segment, our science correspondent Kim Ji-yeon looks into local researchers that have made efforts to turn this into reality.
Treatment using molecular scissors involves taking out nocuous DNA and then inserting the edited DNA back into the host body. Theoretically, the method is expected to be more effective than current treatments, and that it could be used to prevent the occurrence of diseases before they progress and remove the source of the disease instead of tackling the symptoms. However, many hurdles currently exist to apply the method in reality. One of the obstacles involves verifying whether a hazardous DNA has been completely removed and correctly inserted in the host body after it's been edited using the molecular scissors method. To address this issue, a local team of researchers developed a procedure to effectively evaluate the activity of molecular scissors by profiling 11,000 target sequences and coming up with algorithms to predict the effectiveness of new DNA sequences. The technology is expected to significantly reduce the time and cost to verify the effectiveness of molecular scissors used in research. Traditionally, it took a researcher almost a week to look into less than 20 target DNA. Also, the cost to carry out the verification process has not been cheap. It usually costs around $10,000 to verify the effectiveness of several hundred types of edited DNA. To prove the effectiveness of the molecular scissors, researchers would have to survey several thousands of DNA to validate their performance.
We were able to develop a library of guide RNA and target sequence pairs of more than 11,000 DNA sequences during the past year. Using the library, we can verify the effectiveness of the molecular scissors in one single try, reducing costs by more than 1/100th of its original costs.
Another team of researchers at local biotech firm Toolzen addresses the issue of realizing the potential of molecular scissors in medicine by providing quality experimental subjects, specifically genetically edited rats. The rats are different from the currently widely used experimental mice in that their genes have been edited using molecular scissors and not the previous methods that use stem cell research, which can take up to 4 years. Using molecular scissors, it takes less than 4 months. Also, it costs less than 1/5 of what it takes with previous methods.
The rats are bigger in size than experimental mice, making it easier to take MRI or CT images to check physiological reactions. Researchers can draw from the rats several times, whereas the experimental mice die after one blood gathering.
Toolzen plans to commercialize its genetically edited rats starting next year and expand types of experimental subjects using other animal species in the near future. Kim Ji-yeon, Arirang News.